Hi, this is the recording to accompany the slides on the lecture on the heart. So let's begin. This slide gives you the overview of what we're going to cover today. So we're going to start with the basics, followed by the cardiac conduction pathway, intrinsic rhythm, and the ECG, and then with a brief discussion on the cardiac cycle, and then we're going to finish off with heart sounds. So when we look at the basics of the heart, what we can see is that the heart is located in the mediastinum within the thoracic cavity. So if you look over here, this is the thoracic cavity right over here, and the heart is located between the two lungs, right? If you look at the midline, what you can see is that 2/3 of the heart is located to the left of the midline, a third is located to the right. The other thing you can see is when you look at the heart, the bottom border rests on the diaphragm. The bottom border, the lower border is called the apex. The upper border is called the base. And you can see the dorsal surface, the back end of the heart, is located near the vertebral bodies over here, and the front or anterior end of the heart lies just deep to the sternum, the breastbone, and the costal cartilage. The heart is an organ that's the size of your fist, so not very big. And in terms of the weight, you can see here it weighs about 200, so 1/4 kilo for females and just slightly more for males. And it's actually quite an amazing piece of organ, because if you think about the ability of the heart, which is fist-sized, to actually pump blood throughout every single capillary bed throughout your body, it's quite amazing that it's only the size of your fist. When we look at the wall of the heart, what we can see here is it can be divided into 3 distinct layers. So the innermost layer, the one that's in touch with blood, is called the endocardium, and it's a simple squamous epithelium. So simple means 1 layer. Squamous means it's flat. Epithelium, and this endocardium is continuous with the endothelium of blood vessels. The presence of the endocardium also protects the myocardium, which is the muscle part of the heart, from direct contact with the blood. And so if you can see here, the middle layer is the myo. Myo refers to muscle. Cardium refers to heart. And what you can see here is that the myocardial layer of the heart is the thickest layer of the heart, because the function of the heart is to actually contract and push blood out to different parts of the body. So it needs to be thick in order to actually generate enough force to push blood throughout the various blood vessels. The outer layer is called the pericardium, and what you can see here is the pericardium is divided into several different sections. So the outer layer of the pericardium is called the fibrous pericardium, and the inner part of the pericardium is called the serous pericardium. So when we look at the serous pericardium, what we can see here is that the innermost layer, the one that's stuck to the myocardium of the heart, is called the epicardium, and it's a visceral layer of the serous cardium. And then you've got a space called the pericardial cavity that is filled with fluid. And then on the other side of the pericardial cavity, we've got the parietal layer of the serous pericardium. And so what happens is that the fluid over here, there's only about really 10 to 15 mils of fluid that basically reduces the friction generated by the heart as it contracts and relaxes. So you've got this layers of the heart, each of which has its own function in order for the heart to actually work properly. Another part of the heart is the septa. So the septa is the part of the heart that divides, or the walls of the heart that divides the heart into chambers. So you've got the heart over here, And what happens is there are 4 chambers of the heart. So the walls that divide the chambers, that divide the heart into the 4 chambers are called septa. So what we have is we have here the atrioventricular septa that divide the atrium on the top here from the ventricles in the bottom. We've also got the interatrial septum that divides the right atrium from the left atrium, and we've got the interventricular septum that divides the left atrium from the the left ventricle from the right ventricle. Typically, what happens in this is that in a normal heart, I'm just going to draw it again. What happens is that normally there is an opening between each atrium and ventricle, and this allows blood to flow between the atria to the ventricles. And in the fetal heart, there is an opening between the right and the left atria. And what happens is then that allows blood to flow from the right atria to the left atrium, from the right to the left atrium in the fetal heart to bypass the pulmonary circulation. And what happens is when after birth, this hole closes up, and so blood doesn't flow directly from the right to the left atrium, but it goes from the right atrium to the right ventricle and then to the lungs, and then from the left atrium to the left ventricle, and then through the systemic circulation. Normally, there is no hole in between the right and the left ventricles, right? So the holes that are, the openings that are present are the openings between the atria and the ventricles, the opening in the fetal heart between the 2 atria, and there's really no opening between the 2 ventricles. So speaking of which, these are the chambers of the heart. So we've got 2 atria on top, 2 ventricles down the bottom. The atria is smaller than the ventricles. And what happens is that we've got this sort of situation where you've got the right atria, right ventricle, left atria, left ventricle. And what happens is the purpose of the of the atria is to actually pump blood to the ventricles, and then the purpose of the ventricles is to pump blood into the circulation pathway. So the right ventricle pumps blood to the pulmonary circulation, the left ventricle pumps blood to the systemic circulation. We've got auricles attached to the top part of each ventricle, and they can fill with blood and empty into the atria. And then we've got sulcus on the superior external surface of the heart, which contains major coronary blood vessels. And then we've talked about septa before. So when we look at the thickness of these 4 chambers, what we can say is that the myocardium, the muscle layer of both atria, is thinner than that of both ventricles. And that's because in the atria, the purpose of the atria is just to pump blood to the next section, which is the ventricles. And the ventricles are right next to the atria. And then when we look at the thickness of the walls of the ventricles, what we can see is that the myocardium of the right ventricle is thinner than that of the left ventricle. And that's because the right ventricle pumps blood to the pulmonary circulation, whereas the left ventricle pumps blood to the systemic circulation. And we've got more blood vessels in the systemic circulation than we do in the pulmonary circulation. And also the pulmonary circulation is the blood that's pumped to the lungs and back. And the lungs, as we said before, is right next to the heart. So again, a shorter distance, you don't need that much, that thick of a muscle to actually pump blood to the lungs that are next door, compared to the systemic circulation, which can pump blood a much further distance. In between the four chambers, we've got valves. So you can see over here, you've got the right ventricle, right atrium, right ventricle, left atrium, left ventricle. We've got valves in between each atria and ventricle. And these valves, valve A and valve B, are called atrioventricular valves, or AV valves, because they're in between the atria and the ventricles. The one on the right side of the heart is called the tricuspid valve, and the one on the left side of the heart is called the bicuspid valve. And the bi and the tri refers to the number of valves. So the bicuspid valve over here has got the bicuspid has got 2 flaps, and the tricuspid here between the right side of the heart has got 3 flaps. And so that's how they come up with the name. There are also valves between the right ventricle and the pulmonary trunk, as well as between the left ventricle and the aorta. And these are called semilunar valve. And the reason why they're called semilunar valve is because they're shaped like half moons. And so the the semilunar valve in between the right ventricle and the pulmonary trunk is called the pulmonary valve. And similarly, the semilunar valve between the left ventricle and the aorta is called the aortic valve. When we look at how these valves work, what happens is these valves are very, very important in order to direct and permit one-way flow of blood. So these valves open when the pressure in the first compartment is higher than that of the second compartment, because it allows blood to flow down the concentration gradient. So for example, if the pressure in the right atrium is increased and is higher than that of the right ventricles, then what happens is this tricuspid valve will open. And then what happens is when the ventricle starts contracting, what happens is the pressure in the right ventricle will increase. Once the pressure in the right ventricle increases to a level that's higher than that in the right atrium, then this AV valve will close. And the reason for that AV valve to close is that this prevents backflow of blood to the first compartment. Right? Because what you want to do is you want blood to flow in one direction. So you want blood to come in from the superior vena, from the vena cava into the right atrium, from the right atrium into the right ventricle, from the right ventricle into the pulmonary trunk. You don't want it to go back, right? And so the closure of the valves prevent blood from going back to the previous compartment and keeps blood moving along. All right. For the AV valves, what happens is the first compartment is the atria and the second compartment is the ventricles. So the AV valves open when atrial pressure is greater than ventricular pressure and closes when atrial pressure becomes less than ventricular pressure. And so blood doesn't move backwards from the ventricles into the atria. For the semilunar valves, the first compartment is the ventricles, and the second compartment is the great vessels. on the other side of the SL valves. And SL valves then open when ventricular pressure is greater than the pressure in the great vessels to actually push blood from the ventricles out into the great vessels. And then the SL valves close when ventricular pressure drops to a level below that of the of the pressure in the great vessels to prevent backflow of blood back into the ventricles. So you want to keep the flow of the blood moving forward. When both atria and ventricles are relaxed and the AV valves open, blood passively moves from the veins, from the superior and inferior vena cava to the atria and straight to the ventricles. So we'll talk more about that as we go along, right? So what happens is during atrial contraction and ventricular contraction, the AV valves are open and the SL valves are closed. So what happens is you have this format where blood goes straight from the great veins into the right atrium, into the and from the right atrium it goes directly into the right ventricle, and then it stops in the right ventricle because the SL valves are closed, whereas the AV valves are open. And again, from the pulmonary system, it goes into the left atrium, directly into the left ventricle, and then it accumulates in the left ventricle until ventricular contraction happens because the aortic valve is closed and the bicuspid valve is open, right? And this means that blood accumulates in the ventricles until ventricular contraction happens. And when ventricular contraction happens, what happens is if you look at the green line over here, with ventricular contraction, what happens is the ventricular pressure increases. And over here, the SL valve is still closed until the ventricular pressure becomes larger than the pressure in the great vessels. This is the left side of the heart. And so the great vessel in this is the aortic pressure. And once the ventricular pressure is greater than the aortic pressure, then what happens is a semilunar valve opens. And over this part, what happens is blood leaves the ventricles the left ventricle to enter into the aorta. And it keeps on going until the ventricular pressure drops below that of aortic pressure. And when ventricular pressure is less than aortic pressure, then the semilunar valve closes, right? And then what happens is ventricular pressure drops during relaxation, ventricular relaxation over here. And then when ventricular pressure is lower than that of atrial pressure over here, then the AV valve opens, right? So draw a map, draw the picture of the heart to just familiarize yourself with it, because there are a lot of moving parts and a lot of different pressures in different parts of the heart as well as the great vessels, and this is the way that you can actually visualize it in your mind. When we look at the internal features of the ventricles, and again remember the ventricles are the two chambers of the heart that have to pump blood a fair distance. What we can see here is there are three different features that I want to talk about. The first one is the trabeculae carneae. So carneae refers to meat or muscle, and what the trabeculae carneae is if you look right over here, is there are beam-like projections inside the ventricles that help to increase the force of the ventricles when it contracts. And the reason for that is if you increase the force of the ventricles when it contracts, then more blood can leave the ventricles. When more blood leaves the ventricles, more blood can enter into the pulmonary and systemic circulation, and so blood transport increases. The other two features of the ventricles are to do with controlling how the AV valves, the valves between the atria and the ventricles, open and close. So you've got the chordae tendineae, so you can see tendinous cord, and then you've got the papillary muscles. So the papillary muscles here are attached to the AV valves through the help of the chordae tendineae. And what happens is that these two features are contracted when the ventricles contract in order to help to close the AV valves. And the reason is that the AV valves need to close because you don't want this situation where you have the AV valves in between the atria and the ventricles opening up, because during ventricular contraction, you want blood to move out of the semilunar valves. You don't want it to go back into the atria, right? So these two things, the chordae tendineae and the papillary muscles, help to close these AV valves, help to hold these AV valves closed so that blood doesn't go backwards into the atria. So again, more blood leaves into the pulmonary and systemic circulation, prevents blood from going back into the atria. The next two slides talk about the coronary and the nerve supply of the heart. So over here, you've got coronary arteries supplying oxygenated blood to the muscles of the heart. So this is not the blood that goes in the chambers. This is the blood that supplies the thickness of the wall muscle of the chambers. And what you can see here is that the left ventricle receives a large amount of blood from various different arteries, because the left ventricle is the thickest, has got the thickest wall of all the four chambers, right? So we want to actually have an increased blood supply to the left ventricular wall. And then the coronary veins generally follow the path of the coronary arteries. And what happens is blood is collected from various different chambers of the heart. It gets collected from the cardiac veins to the coronary sinus. And from the coronary sinus, it enters into the right atrium in order for it to actually go into the pulmonary circulation to be oxygenated. When we look at the heart muscle and compare it to the skeletal muscle, what you can see here is there are some commonalities as well as some differences. One of the commonalities, similarities between heart muscle and skeletal muscle is that both of them are striated. So striated means that there are bands of light and dark. And so you can see here, this is a band of dark over here. And then in between the bands of dark, you've got a band of light. So again, band of dark, band of dark, band of light, right? So those are similar in both skeletal muscle as well as cardiac muscle. The other thing is that both of them have got T tubules or transverse tubules to aid in the spread of the action potential. Differences in the anatomy of skeletal versus cardiac muscle is that cardiac muscles are smaller in diameter and shorter than skeletal muscles. So they're thinner than skeletal muscle and they're shorter than skeletal muscles. Cardiac muscles also branch freely. So you can see over here, this is one cardiac muscle over here, this is another, and you can see that this cardiac muscle branches, right? And this one does branch theirs too, that one branches there. And what you can see is the junction or the connection between one cardiac muscle to another is through this intercalated disc, right? So you've got this intercalated disc over here that joins one cardiac muscle to another. And what you can see is where one cardiac muscle cell joins another, there are connections. And these connections can include desmosomes and adherens junction to actually help to keep adjacent skeletal muscle cells together. And there are also tight junctions there, which is not shown over here. But the unique part about the skeletal muscle is the presence of these gap junctions. So these gap junctions are like tunnels, and these tunnels help ions to go from one skeletal muscle cell to another very, very quickly, so that there is synchronized contraction within all the myocardial cells of the ventricles and all the myocardial cells of the atria, right? Because you want the ventricles to contract together as one unit, and you want both atria to contract together as a unit. You don't want like a dysfunctional rhythm where maybe the top half of the ventricle contracts before the bottom half and the right contracts before the left. You want both ventricles to contract at the same time, synchronously. In terms of the physiology of the cardiac versus muscle cell, what we can see here is that skeletal muscle has chemical synapse. So remember chemical synapse, where neurotransmitters are released, it binds to receptors, which then open ion channels in the muscle cell. And we also have talked about how in the skeletal muscle, the action potential is relatively short and the contraction is also relatively short. So this is a typical action potential over here, which is very similar to the action potential that we see in muscle cells. And what you can see here is that the voltage rises when sodium ions enter into the cells. And the voltage falls when potassium leaves the cell, right? And then it's got reset. You've got this reset happening over here with sodium, potassium, ATPase channels. So this is a very different profile than what we can see in cardiac muscle. So this is the profile of the action potential in ventricular cardiac muscles. And what we can see here is that we still have influx of sodium leading to a depolarization. We still have potassium leaving to cause a repolarization. But what's unique in the muscle cell is this middle bit in between. And this middle bit in between, called the plateau, is present because what happens is calcium channels are involved. Calcium enters into the cardiomyocytes, the cardiac muscles in the ventricles, and it causes this plateau, right? And so you've got this kind of profile of the cardiac muscle, and because you've got this plateau over here, what happens is the cardiac contraction is slower and is sustained for a longer time. So if you look at this, this is the same time scale in the cardiac muscle as it is in the skeletal muscle. Skeletal muscle contraction only lasts for maybe 50 milliseconds, whereas you can see here the cardiac contraction in the ventricles lasts for a much longer time than that. And then over here, we've got the electrical synapse, where basically the gap junctions allow these ions to actually flow from one cardiomyocyte to another to allow coordinated contraction of the different parts of the heart. In terms of the various types of cardiac muscles that are present in the heart muscles, what we can see here is that most of the muscles in the heart is called cardiomyocytes or myocardiocytes. Like, they're both the same thing. And this myocardiocytes actually contract to help to pump blood throughout the body. So 99% of cells in the myocardium are cardiomyocytes. The other 1% are myocardial conducting cells. So these help to initiate and propagate action potential to help to coordinate the contraction of various parts of the heart, right? If not, the heart is a very large organ, and it needs that coordination between the different parts of the heart, and this coordination is through the myocardial conducting cells. Unique property of the cardiac muscle is it has autorhythmicity. So it can actually initiate an electrical potential, a heart rate that spreads rapidly from cell to cell to target cardiac contraction. And so if you look at this picture on the bottom right-hand side, it's got the different myocardial conducting cells actually highlighted there. So if you look over here, there's 5 different locations that myocardial conducting cells are found in order to actually have a smooth and a coordinated contraction of the heart. And we'll go and look at that over here. So, as I said, myocardial conducting cells form about 1% of the myocardium of the heart muscle, and they form the conduction system of the heart. What happens is this is the sequence that it follows. So the it starts off in the sinoatrial or the SA node and goes from the sinoatrial node to the AV node. From the AV node, it goes to the AV atrioventricular bundle. From that, it goes to the right and left bundle branches. From that, it goes to the Purkinje fibers. Now, each of these sections of the cardiac conduction pathway can actually generate its own rhythm. But what you can see is as it goes further and further from the SA node, the rhythm that it can generate becomes slower and slower, right? So this is not a problem because what happens in the heart is that the fastest rhythm actually governs or sets the pace for the cardiac contractions. And so if your whole cardiac conduction pathway is working, what happens is that the SA node sets the rhythm at 80 to 100 beats a minute. If there is something wrong with the SA node, then what happens is the AV node then generates the rhythm, and it generates the rhythm at a slower pace at 40 to 60 beats per minute. If the AV node is faulty or what have you, or if the connection between the AV node and the AV bundle is damaged, then what happens is the AV bundle sets its the rhythm of cardiac contraction at 30 to 40 beats a minute, and so on and so forth. So most of the time in a healthy heart, what happens is the SA node sets that the rhythm of cardiac contractions at about 80 to 100 beats a minute if there is no input from either the nervous or endocrine system, right? So this is, if you actually take out the heart in its entirety, what happens is the heart will beat at 80 to 100 beats a minute. But our heart is in our chest, our heart is linked to the nervous system and the endocrine system. So those two systems help to affect the speed of cardiac conduction. All right, so in terms of the sequence, we start over here, point number 1. So point number 1, cardiac conduction system is at rest. And then what happens is that you've got these pacemaker cells in the SA node over here that starts the action potential. So it starts it at 80 to 100 beats a minute at rest without any endocrine or neural intervention. And then from the SA node, it travels to the AV node over here. And then what happens is the both atria contract at the same time and pushes, and the contraction of the atria then actively pumps blood from the atria to the ventricles. And then at that point in time, what happens is that the electrical activity travels from the AV node then to the AV bundle over here, to the bundle branches, and then to the Purkinje fibers on the outside of the ventricles. And after that happens, then what happens is the two ventricles contract in a coordinated fashion. And when the two ventricles contract in a coordinated fashion, then you've got blood leaving the ventricles when the pressure in the ventricles is greater than that of the great vessels, so that the SL valve opens. When we look at what happens, so this is what happens in the SA node. Remember, the SA node is the start of the pacemaker. And so what happens is you've got this typical format where the membrane potential increases, depolarizes due to sodium entering into the cell. And then at some point, calcium channel opens. And then after that, so that when calcium channel opens, calcium ion enters into the cell, and then potassium channel opens to allow potassium to leave the cell. And when potassium leaves the cell, then you've got this repolarization happening where membrane potential drops. Unlike skeletal muscles and neurons, cardiac conductive cells do not have a stable resting membrane potential. So if you look at this, what happens is after it repolarizes, it doesn't stay flat, right? The membrane potential after repolarization is flat until another action potential hits. In the SA node, you don't have that stable resting potential, and so another action potential can start because of this instability of resting potential and because of the autorhythmicity of the conducting cells in the SA node. When we look at the electrical activity in ventricular muscles, what you can see here is, again, phase one, you've got sodium ions entering into the muscle cells. You've got this plateau when calcium channels open and calcium enters into the cell, and then the repolarization occurs when potassium channels open and potassium leaves the cell. When we look at the refractory period, remember, a refractory period is when a second action potential can either be generated if there's a strong enough force, in which case it's a relative refractory period, or when no second action potential can be generated regardless of the electrical stimulus, in which case it's an absolute refractory period. And so what you can see here is that the refractory period is actually quite long in the myocardiocytes compared to in the skeletal muscle, right? And that's because, especially in the ventricular muscles, it takes a while for the ventricles to actually contract and to reach a pressure that blood will actually start leaving the heart to enter into the great vessels. And so that takes time. And so you don't want the ventricles to contract so quickly, one after another, because then that means that not enough blood will actually leave the ventricles. So you want a nice slow ventricular contraction in order to allow blood to actually leave the ventricles. ECG, or electrocardiogram, is a chart which basically records the heart activities. And so this is done by placing electrodes on the subjects. So this is the electrodes and the placements. Again, you don't have to remember where they're located, just that it's located in different parts of the body. And then what happens is that allows a voltage changes of the heart to be actually recorded. So again, this is just a GIF which shows you the different sections of the ECG and how they correspond to systole. So systole means contraction, diastole means relaxation. So how each part of the ECG correspond to either atrial systole, ventricular systole, atrial diastole, ventricular diastole. Right? So we'll talk about that more as we actually go through here. So there's three main phases in an ECG. The first one is a small P wave. So the small P wave is right over here at the start, and that is caused by atrial depolarization. And after the small P wave happens, you've got atrial contraction. That's followed by this QRS complex, which is larger, and two things happen as a result of the QRS complex. One is that because it happens after the small P wave, because it happens after contraction of the atria, what happens in the QRS complex is that the atria relax or repolarize. At the same time as the atria repolarizing and relaxing, what happens is you've got ventricular depolarization, which is followed by ventricular contraction, right? So at the same time as ventricular contraction happening, the atria relax. And then the third part of this is the T wave. And during the T wave, the ventricles repolarize, and that then leads to relaxation of the ventricles, right? We've got over here just the different intervals and segments. We're not going to go much into that, but when we talk about altered physiology or pathophysiology, these segments and intervals are clinically relevant because if they're lengthened or shortened, that may be indicative of something wrong with the heart. And you can, it's actually a nice non-invasive way of trying to figure out what's going on in the heart. Last thing, or second to last thing, we're going to talk about the cardiac cycle. And so the cardiac cycle is one whole cycle which starts with atrial contraction and ends with ventricular relaxation. So you've got these two graphs over here, which depict the same thing in different ways. So when you go over here, what you can see here is this one over here depicts pressure in the aorta. The one that is right below it that goes up to aortic pressure is the pressure in the left ventricle. The one that basically hangs around down the bottom shows you the pressure in the left atrium. So this one over here, as well as this blue line way over here, right? And then you've got over here what's happening in the ventricles and the atria, as well as what's happening in the ECG. One reason for including this graph over here is that we have an understanding of when the AV valve opens and closes and when the SL valve, or in this case the aortic valve, open and close. We also have over here when the atria contract, systole, and when they relax, diastole, as well as what happens with ventricular contraction, systole, and ventricular relaxation in diastole, right? The graph over here gives you some of the same information with the added bonus of this one over here, which shows you what happens in the ventricles in terms of how much blood is actually found in it. So you can see over here that ventricular filling happens during ventricular relaxation. So you can see here blood flows into the ventricles over here. And then when the atria contract, you've got another little spurt of blood entering into the ventricles. And then during, when the ventricles, when the atria contract, you got another little bit of blood entering into the ventricles. And then when the ventricles start contracting, you've got this small area over here where ventricular pressure increases, but there's no blood leaving the ventricles. And this period over here is called isovolumetric contraction, where basically the pressure increases, but at this point, the pressure in the ventricles, it's still lower than the pressure in the aorta, so the SL valves are closed. And then once the pressure in the ventricles are higher than that in the aorta, the SL valve opens, blood leaves the ventricles, and so you've got this drop in blood volume in the ventricles until the pressure in the ventricles is lower than that in the aorta, and then the SL valves close, right? And so you've got there. Last thing in this is that you've got this S1 and S2. S1 and S2. And this S1 and S2 are the two sounds that are very commonly heard through a stethoscope. And sometimes they're called S1, S2. Sometimes they're called lub and dub because it's lub dub, lub dub, lub dub. And this S1, the lub and the lub dub, is a sound that's made when the AV valves close. And the S2 or the dub in the lub dub is made when the SL valves close. Right? So there's sounds that are made when the valves close. There's no sound when the valve opens, because like when you open the door, there's really no sound. When you slam the door shut, you've got that sound, right? So the lub in the lub dub is S1 when the AV valves close, and the dub in the lub dub is S2 when the SL valves closes. Just a couple of other things. As I said, diastole is a period of relaxation. Systole is equivalent to a period of contraction. In diastole, blood can move between chambers, but it's passive filling, whereas in systole, it's active pumping of blood from one chamber to the other. As I've said numerous times, what happens is both right and left atria will contract and relax at the same time, and both right and left ventricles will contract and relax at the same time. The atria and the ventricles undergo contraction and relaxation at different times in order to actually move blood efficiently and effectively from the great veins to the atria, to the ventricles, and to the great vessels, right? Blood, as I said, moves from an area of higher pressure to one of lower pressure, and so that's when the contraction comes in. When you contract, it actually increases pressure in order to try to move blood to the next chamber. The next slide is very, very busy, and the reason why it's very, very busy is I've tried to actually have it all in one slide. And so if you look at these diagrams at the top, first of all, what you can see is that the first diagram over here is identical to this diagram over here, because I wanted to depict that this is a cycle, right? So it goes from this to this, to this, to this, to this, to this, and then all the way back to the beginning, right? So I've deliberately put it over there. One of the things in order to actually read the diagram is this. So anytime you've got these black arrows that basically show you whether the chambers are relaxing or contracting. So a black arrow pointing to inside the chamber shows contraction. So in this one, you show that the ventricles contract, right? So the ventricles are contracting in this diagram, and the atria are relaxing because the black arrows point outwards away from the heart, right? Over here, the next part, you still got the ventricles contracting and the atria are still relaxing. The difference between this and this is that the pressure in the ventricles in, say, diagram A is not higher than the pressure in the great vessels. So you've got the SL valves over here. And what you can see is you've got ventricular contraction, but there's no movement of blood out of the ventricles. Whereas in this picture over here, B, you've got movement of blood from the ventricles, from the left ventricle into the systemic circulation, and movement of blood in the right ventricle into the pulmonary trunk, right? And so let's go and go through this. So at the start of the cardiac cycle, what happens is both ventricles and atria are relaxed. And when both ventricles and atria are relaxed, what happens is that the AV valves are open as the atrial pressure becomes higher than the ventricular pressure, and the SL valves are closed because the pressure in the ventricles is lower than that of the aorta or the pulmonary trunk. And so what happens is it allows blood to go from the veins into the atria directly into the ventricles on both sides of the heart. And then, so this happens when there's nothing happening in the ECG. And then atrial systole starts with the P wave of the ECG. And what happens over here is that the atria contract. And when the atria contract, pressure in the atria increases to actively push blood into the ventricles. And so what happens is most of the blood enters during ventricular and atrial relaxation, but 20 to 30% of the blood moves from the atria to the ventricles during atrial contraction. The next phase after that is when the atria start relaxing and the ventricles start contracting, right? So when the ventricles start contracting, what happens is the pressure in the ventricles gets bigger than that of the atria, so the AV valve close. The pressure in the ventricle in the first instance is still less than the pressure in the great vessels, and so the SL valves don't open yet. The SL valves then open only when the pressure in the ventricles is greater than that of the great vessels and blood starts leaving the ventricles. So 70 to 80 mils of blood leaves each ventricle actively, leaving 50 to 60 mils of blood left. And this all happens during the QRS complex. At the end of ventricular relaxation or ventricular contraction or ventricular systole, what happens is relaxation follows. And so the last phase is ventricular diastole. When the ventricles are relaxed, atria are already relaxed. And so you've got it over here, ventricles relax. And then when ventricular pressure drops so that atrial pressure is higher, then it opens up the AV valves. And when the AV valve opens, then blood can actually go from the veins to the atria directly into the ventricles, right? And at this point, the SL valves are closed because ventricular pressure is lower than that of the pressure on the other side of the SL valves in the great vessels. And this is during the T wave. So anyway, we'll cover this more in greater detail, I'm sure. But just go through this, look at the diagrams, figure out what's happening in both the atria and the ventricles, in both sets of valves, to actually get a fuller picture of how it all works together. There's some animations here just to actually show you that the coordination of the two atria happens, contraction and relaxation of the two atria happens synchronously. Contraction and relaxation of the two ventricles happens synchronously as well. And then this one, I'm hoping to show that cardiac conduction is coordinated with contraction and relaxation of the four chambers of the heart, right? So signal for contraction starts in the sinoatrial node, and that causes as that's happening, you've got relaxation of all four chambers, and then when the atria contract, then it actively pumps blood from the atria to the ventricles. And then as the signal is transmitted down the bundle of His around the Purkinje fibers, then what happens is the ventricles start to contract. And when the ventricular pressure is higher than that of the pressure of the aorta and the pulmonary trunk, then the SL valves open and blood leaves the ventricles to enter into the pulmonary and systemic circulation. And then the last part are the heart sounds, which I've covered before. So you've got the lub-dub, lub-dub, lub-dub. And you see over here that the first sound, lub, is due to the AV valves closing. The second sound, dub, is due to the semilunar valves closing. There are two other valve sounds, but these two valve sounds are very rare in healthy individuals. If it can be heard with a stethoscope, it may indicate some sort of heart failure because it's the S3 valve is the sound of blood moving back into the atria, and the S4 sound is made when blood moves from the atria into a stiff or a hypertrophic ventricle. Over here, this diagram over here shows that you listen to the heart sounds using a stethoscope, and if you've ever wondered why when you go to the doctor, the doctor or healthcare professional moves the stethoscope around, it's to actually to hear how the different valves actually function. So placing the stethoscope here gives you the aortic component of the SL valve of the left ventricle. The same with this is SL valve in the right ventricle, and then you've got the tricuspid in the right ventricle, and then the bicuspid in the left ventricle. Oh, it's the opposite around. Bicuspid in the left ventricle, tricuspid in the right ventricle. So again, you next time you go to the doctor or you see someone and they're checking around in different parts, then you know what it is. Okay, that's it for now.